Meiosis. The overview meiosis is that it is when a single cell divides to form 4 genetically different cells, which are haploid cells. They have 23 chromosomes. They are formed through reduction division because they have half the amount of chromosomes. This process has 2 cycles, unlike mitosis, which just has 1. And this is important because of sexual reproduction, which creates gamete cells, and for genetic variation, which is important for evolution. So in meiosis 1, the first phase is interphase, and that is when the cell grows and copies all of its chromosomes. It prepares for division, and it is a diploid cell. So we have the centrioles, which are cylindrical organelles near the nucleus, and they're involved in the development of spindle fibers. The nucleolus is a small dense structure in the nucleus during interphase. Chromatin is what the chromosomes are composed of. The nuclear envelope holds the chromosomes, and the plasma membrane protects the cell. In prophase 1, the chromosomes condense and pair up, and crossing over occurs. Crossing over is the exchange of genes between homologous chromosomes to increase genetic variation, and synapsis is the process of chromosomes pairing. Metaphase 2 is the second part, and this is when spindle microtubules move homologous pairs to the middle of the cell, and bivalents align on the equator. Bivalent is the pairing together of homologous chromosomes. Chiasmata is the point where crossing over occurs on the chromosomes, and this happens in prophase 1. Homologous chromosomes are not identical, but they have the same genes in the same order, but the alleles for that trait are not always the same. In anaphase 1, homologous pairs are separated with 1 chromosome of each pair moving to a pole, and random orientation occurs in this phase. Random orientation is when the orientation of homologous chromosomes prior to separation is random. In telophase 2, the chromosomes uncoil, and there are now 2 haploid cells, and cytokinesis occurs, which splits the diploid cell into 2 cells. And prophase 2, this is the start of the second cycle, is first of all, the chromosomes condense in each of the 2 cells. In metaphase 1, the spindle microtubules align and align the chromosomes to the center of the cell. In anaphase 1, centromeres separate and chromatids are moved to opposite poles.
Anaphase 2.
That's what I meant. In telophase 2, chromatids reach opposite poles, the nuclear envelope fors, and cytokinesis occurs, which splits the 2 cells into now there are 4 genetically different haploid cells. And finally, there's cytokinesis, which is the division of the cells. This occurs after telophase 1 and 2. And yes, here are our references.
Thank you for watching.